Ask me.
Spermatogenesis. Okay. So we discussed about the spermatogenesis in the first lecture. We start from the very basic. Okay. That what is embryology? Embryology means formation, development, and maturation of embryo. Okay. Then the whole pregnancy, 9 months, we divide in 3 periods. That is pre-embryonic mean where the germ cells are formed. Okay? Germ cell, ectoderm, mesoderm, and endoderm. And in the embryonic period, that is organogenesis occur. Organogenesis from that germinal layers, different system are formed. That layers are known as that formation of different system is known as organogenesis. And then the fetal period. In fetal period, that organ which is formed in the embryonic period become developed. Okay, they develop, become mature. Okay, then we start about the gametogenesis. So there are two types. One is spermatogenesis, another is oogenesis. Spermatogenesis mean formation of sperm. Gametogenesis mean formation of the ovum. Okay. Now after that, from where that spermatogenesis and gametogenesis occurs in the epiblast. In the epiblast, primordial germ cell develops, which migrates from the epiblast to the wall of yolk sac, and from the wall of yolk sac that primordial germ cell migrate to the genital ridge, and from genital ridge is that part of the embryo from where male and female reproductive system develop. Now from the primordial germ cell in the genital ridge, primordial germ cell develop into oogonium and spermatogonium. Okay. Now spermatogonium undergo mitosis and it develop into two types. One is black, one is pale and another is dark. Okay. One is dark. Dark is also known as what? That is type A, and pale is type B. From that type B, spermatogenesis is occur. And through mitosis, that type P spermatogonium develop into primary spermatocyte. After meiosis 1, primary spermatocyte develop into secondary spermatocyte. After meiosis 2, that is developed into spermatid. Okay, and then in spermatid spermiogenesis occur and that develop into mature sperm or spermatozoa. Okay, so the conversion of spermatid into spermatozoa is known as spermiogenesis. Okay, spermatozoa is present in the form of what? In the form of normal cell, that is Golgi apparatus, endoplasmic reticulum, okay, and nucleus and centrioles. From one centriole, that neck of the sperm develop. From another centriole, tail of the sperm develop, and from the mitochondria, middle piece of the sperm develop. From the nucleus, head of the sperm develop, and from the Golgi apparatus, that acrosomal cape is developed. Now spermatogenesis occur in the Sertoli cell of the testes. Sertoli cell is present in the seminiferous tubule. Now the spermatozoa or the sperm which is released from the Sertoli cell is known as spermiation. Okay, so there is three words: spermatogenesis, spermiogenesis, and spermiation. Spermatogenesis means formation of the sperm. Okay? Spermiation means release of the sperm from the holy cells. Okay? And that spermiogenesis, conversion of spermatid into spermatozoa. Okay? Now, from the seminiferous tubule, the sperm go to the red testis. From red testis, it go to the epididymis. In the epididymis, it get mature. Okay? And from that get motile and stored in the epididymis. From the epididymis, it will go to the vas deferens. From vas deferens, it will go to the ejaculatory duct. From the prostate gland, semen will mix to the sperm and then they will go to the urethra and ejaculate out. That is about the previous class. Hormone needed for the spermatogenesis is FSH, LH and testosterone. बेशक FSH and LH मत बोलो, लेकिन testosterone आपको याद होना चाहिए. Clear? Now, today we will start from the oogenesis. From the oogenesis. Now, yes, duration needed for spermatogenesis is from 72 days to 74 hours, days, and the lifespan of the sperm is from 48 hours to 72 hours. Okay. Next is mitosis and meiosis. Now, we will start from this oogenesis. Now, oogenesis means formation of the oogenesis. Oogenesis means formation of the womb. Formation of the womb. Now, oogenesis is started when the female baby, is still in the uterus of the mother. So, it is started in the intrauterine life. It is started in the intrauterine life. Okay. Now, the oogonium is undergo mitosis process. That is mitosis process. Through mitosis, oogonium is converted into primary oocyte. Okay, primary oocyte. Now, you people know that mitosis, in the mitosis, the number of chromosome will be remain same. There is no reduction of the chromosome. Now, this primary oocyte undergo the first meiotic division, first meiosis. Okay. Now, this is the question. This one is the question, that first meiosis start before birth. It start before birth. Okay, before birth, but that meiosis 1 arrested in the, that's arrested mean stop in prophase 1. Stop in prophase 1. Okay, and that is the prophase, sub-stage of the prophase, diplotin stage. Okay, so before birth, what are the factor, what are the conditions which occur before the birth in the oogenesis? That is mitosis. After that, meiosis 1 start, but that meiosis 1 is arrested in phase 1, diplotin stage. Okay, prophase 1, diplotin stage. Now, this after birth, what will happen? After birth up to puberty, this primary oocyte will be arrested in the prophase, but at the time of puberty, this first meiosis division will be completed. First meiosis division will be completed, and primary oocyte will change into secondary oocyte and first polar body. Okay, first polar body. half the chromosome will go to first polar body and half the chromosome will go to the secondary oocyte. Now secondary oocyte again undergo meiosis 2 because meiosis have two phases, meiosis 1 and meiosis 2. It will start meiosis 2 but again it will arrested in the metaphase of meiosis 2. Okay, so these are the highlighted question that first meiosis will be stopped at the phase of prophase, and the prophase that is diplotin stage, and the second second meiosis will be arrested in the metaphase. Okay, now second meiosis will be completed or not, it depends upon the availability of the sperm. It depends upon the availability of the sperm. If the sperm is available, second meiosis division will be completed. But if the sperm is not available, second meiosis division will not be completed. Okay, now sperm, and in the front that I read that is what? That plus. Okay, so mean that sperm is available, so it will complete meiosis 2. It will complete meiosis 2 just before the fertilization. Just before the fertilization, first meiosis division will be completed. Okay, and one and second polar body will be formed. Second polar body will be formed. Second polar body mean that there is an active X chromosome. One chromosome is active, another is inactive. That is known as polar body. And if the sperm is not available, then it will persist as a corpus luteum. It will persist as a corpus luteum. Corpus luteum means that it will shrink. Okay, it will persist as a mean second meiosis division will be not completed. Okay, if the sperm is not available, and it will go with the menstrual bleed as a corpus albicans. Okay, it will convert to corpus luteum, and corpus luteum undergo through apoptosis. Okay, death of the corpus luteum occur, and that is changed into corpus albicans. Clear? And every month there is menstrual bleed, so with that they will go out. Clear? One is apoptosis and another is necrosis. Apoptosis means physiological cell death. Okay, and necrosis means pathological cell death. We will discuss that in general embryology. Okay, now so spermatogenesis and oogenesis is occur. Now we will go for the fertilization in first week. That what will happen if the sperm is available? Okay, if the sperm is available, then what will happen if a womb is fertilized? If a womb is fertilized, ये बोर्ड सबको दिख रही है? Okay. Now the female reproductive system. Okay, this is the female reproductive system. Clear? सबको दिखाई दे रही है या नहीं? This is the female reproductive system. Here is ovary. Okay, there are two ovaries. Okay, now this is fundus. This is uterus. This is uterus. It is the fundus part. Fundus. Okay, then this is ovary. Ovary, there are two ovaries. Okay, it is stabilized by some ligaments. Okay, this is fallopian tube. Fallopian tube. Okay, this is body of the uterus. Body of the uterus. Then cervix. Okay, and in the last, this is vaginal canal. Okay, now every monthly cycle there is one ovum released from each ovary. Clear? From each ovary one ovum is release. Now that is known as ovulation. That is known as release of the release of the womb from the ovary. This is fallopian tube. Okay. This is the fimbriae part. Fimbriae is finger-like projection. Finger-like projection. The second part is isthmus. Okay, that is ampulla. Fimbriae, ampulla, and the last part is infundibulum. Okay, so fallopian tube have 4 parts. One is fimbriae, another isthmus, ampulla, and infundibulum. Okay, now this fallopian tube have finger-like projection from these endothelial cells. These are known as cilia. Okay, these are cilia. Now at every monthly cycle, one womb will be released. Okay, now these are finger-like projection, so this fimbriae will catch that womb. Okay, will catch that womb, and through this cilia is in the form of continuous movement. So it will push this womb up to the ampulla. Okay, and from here sperm, so it will go to the fallopian tube up to the ampulla. So the question, what is the main site? What is the main site for fertilization? What is the site of fertilization? That is ampulla of the fallopian tube. Okay, fertilization occur in the ampulla of fallopian tube. Okay, now when the sperm reach here, only the nucleus will enter to the womb. So half chromosome in the womb and half chromosome from the sperm. Okay, now if this half chromosome is X chromosome, then the baby is female, and if this half chromosome from the sperm side that is Y, then the baby is male. Okay, now this fertilized womb have 2 layers around it. Okay, it have 2 layers. You will study in the gynae. Okay, that the maturation, there are some follicles around it. This womb is developed inside some follicle, that is primary follicle, secondary follicle, and Graafian follicle or tertiary follicle. Okay, now for that we need FSH. Okay, so Graafian follicle. Now from that Graafian follicle, it can take 2 layers of cell around this womb. One is corona radiata and another is zona pellucida. What is the function of Okay, this fertilized womb will undergo the process of mitosis. Okay, mitosis will start. So one fertilized womb will change into 2 cells. Okay, then it will change into 4 cells. Okay, after that it will go into 8 cells. Okay, 8 cells, and then 8 will change into 16 cell stage. Now 16 cell stage of the fertilized womb is known as morula. Okay, that is morula, but that is still surrounded by zona pellucida and corona radiata. What is the function of zona pellucida and corona radiata? It will never ever allow another species sperm to enter into the womb. Okay, enter into the womb. And another second function, it will never ever allow this fertilized womb to implanted in an abnormal area. Okay, for the implantation there is specific site where this fertilized womb will be implanted. Okay, now 16 cell stage is known as marula. Okay, marula. Now it will change into 32. Okay, that 32 cell stage is known as what? That is late marula. Okay, late marula. And the 64 cell stage, at the 64 cell stage, this zona pellucida and corona radiata will be dissolved by the uterus. Here the fluid will dissolve this layer. Okay, and the fluid present here will start entering into this 64 cell stage. Okay, so what will happen when this fluid enter inside this 64 cell stage? It will become blastocyst. Now how blastocyst look like? Okay, that how the structure of blastocyst is look like. Okay, now this is like this. These are 64 cell stage. 64 cell stage. Zona pellucida and corona radiata is dissolved. Okay, that is dissolved. Now the fluid will enter into the cell. Now what will happen? These fluid, uterine fluid, will push. Okay, will push the cell into one side like this. The cell will push to the one side. Okay, and one layer of the cell will be remain like this. Okay, so the cavity. Okay, the cavity is formed. This mass which is pushed toward one side, that is inner cell mass. Inner cell mass. And another, this is outer cell mass. Okay, and this structure, this is known as, these are blast cell, and this is cavity. Cavity in the medical language, cyst. Okay, we call cyst. So this is blastocyst. This is blastocyst, and this blastocyst will come and implanted in the wall of the endometrium, in the uterus, in the body. Okay, it will implanted in the body of the uterus. Okay, it will implanted in the body of the uterus. Clear? This is like that. See. Okay, now this is fertilized womb. Okay, sperm just entered the nucleus, that is zygote. Fertilized womb is known as zygote. That is changed into 16 cell stage, that is marula. Okay, marula. And the fourth day, that will change into the late marula or advanced marula. Okay, then it will, the outer layers will be dissolved. Now look, this cavity, inner cell mass and outer cell mass, this is known as blastocyst, and blastocyst is going to be implanted. Okay, it will implanted in the body of the uterus. Okay, so the time when the and where the implantation will start at the 6th day of fertilization. Okay, it will start at the 6th day of fertilization, and implantation will complete at the 10th day of fertilization. See, stage of implantation. कौन सा स्टेज हमारे साथ इम्प्लांट होता है फर्टिलाइज्ड वूम का? That is blastocyst. Implantation begin at 6th day, implantation completed at 10th day, and site of implantation, junction of fundus and body of the uterus on the posterior wall. This is the exact location where the implantation occurs. Now, if this implantation occurs somewhere else, site of the implantation. भाई मुझसे फिर एग्जाम में मैं किसी का सिफारिश नहीं मानता, ओके? और मैं हूँ भी आप लोग की बदकिस्मती है पठान, एक बात के साथ हम पढ़ जाए ना तो फिर नहीं है, फिर ये हमारी जेनेटिकल इशू है, इसमें हम कुछ नहीं कर सकते। तो मैं सब आप अपने सीनियर से पूछ लेना, मैंने अभी तक किसी को कुछ भी नहीं बोला है, ना मैंने कभी बच्चों को तंग किया हुआ है, लेकिन जिनको किया हुआ है ना उनसे जाके पूछ लेना, 3-4 बच्चों को किया हुआ है, सही है? तो that is the exact location. Now, if this is implanted somewhere else, that is known as ectopic pregnancy. That is ectopic pregnancy. Now, what is the most common site of ectopic pregnancy? Most common site of ectopic pregnancy, that is uterine fallopian tube. Okay? Most common site of ectopic pregnancy is fallopian tube. And another, what is the least common site of ectopic pregnancy? Least common, सबसे कम, most common, सबसे ज़्यादा. So least common is ovary. That is ovary. Okay, that is ovary. Now this implanted womb, that is, now what will happen in the first week of fertilization? What will happen in the first week of fertilization? यहाँ तक किसी का कोई क्वेश्चन है? हाँ? Yes, sir. That is ectopic pregnancy. Yes? No, that is continue, but that the ectopic pregnancy भाई बोल रहे हैं कि अगर ये जो फर्टिलाइज्ड वुम है ये हमारे साथ किसी और जाके स्टिक होता है, तो इसको हम यही मैं बोल रहा हूँ कि अगर ये हमारे साथ कहाँ पे किसी और जगह पे इम्प्लांटेड होता है एक्सेप्ट द बॉडी एंड द पंडस जंक्शन एट द पोस्टीरियर वॉल, अगर यहाँ से जाके किसी और जगह पे इम्प्लांट होता है, दैट इज एक्टोपिक प्रेगनेंसी. And the most common site of ectopic pregnancy is fallopian tube, and the least common is ovary. Okay? Now, of course, continue as ये सही है. But after at the last stage, in the end stage, maybe we will go for C-section. Okay? और जो बच्चे C-section से होते हैं, सही है, उसको सारी उम्र पर complication होती है. क्योंकि उनके लंग्स immature रह जाते हैं, immature, okay, because of there is no stress hormone, okay, हम स्ट्रेस को रिमूव करते हैं, अगर हम सी सेक्शन करते हैं, सही है, उसकी वजह से कुछ स्ट्रेस हार्मोन है कि वो रिलीज नहीं होते हैं, okay. Now that is about the first week of fertilization. Then what is happen in the first week of fertilization? First week fertilization, the blastocyst is formed and that is implanted in the wall of the uterus. Now second week किसी का कोई question है? Any question? Okay. Now this what will happen in the second week of fertilization with this this blastocyst? Okay. What will happen? Now in the second week, everything will become 2-2-2. Okay? The rules, follow the rules of 2-2-2. Okay? अभी बताता हूँ कि क्या होता है. आप लोग जितना शोर करोगे ना, मैंने जितना अपने माइंड में किया हुआ होता है मैं वही पूरा करूंगा. ये जो जर्नल एम्ब्रियोलॉजी है, ये 3 लेक्चर में खत्म करना है तो खत्म करना है. लेक्चर 10 बजे तक पहुंच रहा है, 11 बजे तक पहुंच रहा है. आपके पेरेंट्स को मसला है, इंस्टीट्यूट को मसला है, आपको मसला है, वो आप लोग का हेडेक है, मेरा नहीं. सही है? आप लोग शुगल मस्ती के लिए आए हो तो इधर ना आते, किसी अच्छी कंट्री में चले जाए. ओके. नाउ blastocyst. One is like this, outer cell mass. Outer cell mass. This is blastocyst. Outer cell mass is formed. This is the outermost cell layer. Now, one is inner cell mass. This is inner cell mass. Okay, this is inner cell mass, and this is the cavity. This is the cavity. Okay. Now, what will happen with this blastocyst? Now, first, what are the changes which occur in the inner cell mass? Okay, and what are the changes which occur in the outer cell mass? Okay, in the second week, this inner cell mass, this lower layer of the cell will take this like a structure of cuboidal cell. A layer of cuboidal cell will form from these layers. Okay, cuboidal cell layer is formed from this below layer of the inner cell mass, and this is known as what? This is known as hypoblast. मैंने आप लोग को बोला था कि इपीब्लास्ट था, सही है? वो इपीब्लास्ट कहां से आया था? अब सुन ले, ये हाइपोब्लास्ट बन गया. ओके, and the remaining cell will become columnar structure. That is columnar structure. This layer of columnar structure is known as epiblast. Clear? Now, this hypoblast is also known as primitive endoderm. Mean in the future this hypoblast will be developed into endoderm. That's why it is known as primitive endoderm. And this epiblast is known as primitive ectoderm. Mean in the future it will develop into what? Ectoderm. Now, this cell, inner cell mass, develop two layers. Yes, two layers. One is epiblast, another is hypoblast. Hypoblast are cuboidal shape, okay, and epiblast are columnar shape. Okay. Now, from this epiblast, cell are developed and go and form a layer like this. Okay, this is what? This is amniogenic cell layer. Amniogenic cell layer. So this is amniogenic cell. From where these amniogenic cell will come? That is from epiblast. From the epiblast. So this cavity is formed. This is known as amniotic cavity. Okay. And this cell will develop a layer downward from this hypoblast. Okay. From the hypoblast, this cavity is known as yolk sac, and this outer cell mass, what will happen to this? This cell, some of it will develop into two layers. One layer will be in arranged form, mean it will form like this. In the arranged form, mean we will differentiate every cell. One layer is like that. Okay, and one layer is formed from these layer. Okay, from the which we will never differentiate the nucleus, the cell membrane, that is irregular layer. Okay, this outer cell mass is also known as trophoblast. That is trophoblast. So from the trophoblast, outer cell mass or trophoblast is the same thing. It will develop into two layers of the cell around the embryo. One is irregular. We never differentiate the cell membrane, the nucleus, that which nucleus is regarded to which cell. and which membrane is regarded to which cell, and another is proper layer, well arranged cell. This irregular cell is known as syncytiotropoblast. Syncytiotropoblast. And the inner one is cytotropoblast. Cytotropoblast. What is the importance? This syncytiotropoblast will release beta HCG. And in the start, it will appear in the urine, and we will check pregnancy from this beta HCG, that pregnancy is start or not. Okay, that is beta HCG is coming from syncytiotropoblast. Now, same like this. This structure is formed. This is it will be like this. This is outer cell mass. Okay, it will form tropoblast, and then that is inner cell mass. So inner cell mass, one is developed into hypoblast or primitive endoderm. That only layer of the inner cell mass develop into a cuboidal shaped cells known as hypoblast, which will develop into endoderm. That's why it is known as primitive endoderm. Another, this outer cell mass or tropoblast will develop into cytotropoblast and syncytiotropoblast. In the cytotropoblast will depend cell with cell membrane and nucleus, and in syncytiotropoblast no proper cell membrane and multiple nucleus will be there. Okay, ये लिकुनाई में explain करता हूँ कि what is this. Now from the remaining this in the second diagram, in the second diagram, okay, that columnar cell will be formed from the remaining inner cell mass. That is what? That is epiblast. Okay, the columnar cell layer is formed from remaining inner cell mass known as epiblast or primitive ectoderm. Okay, and that is the tropoblast, amniotic cavity, and that is yolk sac. Okay, the yolk sac is surrounded by the membrane that is known as Husserl membrane. Okay, Husserl membrane. Clear हो गया? It is also known as exocoelomic membrane. Okay, this layer of the cell is derived from the proliferation of what? From these hypoblast cell, amniogenic cell, epiblast cell are proliferate and form these amniogenic cell. Clear हो गया? यहाँ तक clear है? कोई confusion, कोई question है? This is so. Two cavities are formed, two layers are formed. Okay, that is tropoblast and epiblast. That is epiblast and hypoblast, amniotic cavity and yolk sac, and then that is layers, cytotropoblast and syncytiotropoblast. यहाँ तक क्लियर है? किसी का कोई क्वेश्चन है? जी बेटा.
Sir, as you said, the cell mass is developed into two layers. One
Yes.
is proper layer,
Proper.
cytotropoblast, and second one is syncytiotropoblast.
Yes.
Sir, which one release beta HCG?
The syncytiotropoblast.
It is used to diagnose the pregnancy.
Yes.
And
In the
sir-
early stage.
Okay, and sir, one more doubt. Amniogenic cells, which is developed from epiblast, is surrounded by which cells, sir?
That surround the amniotic cavity.
Okay.
That surround, proliferate and surround the like this. It will proliferate, start from here and form a layer around this cavity that is known as amniotic cavity. Clear? Any question? Clear?